The overall goal of this procedure is to quantify the maximum potential rates at which extracellular enzymes in soils degrade their target substrates. This is accomplished by first combining field moist soil and buffer in a blender, and then pipetting the mixed samples into a deep well sample plate and deep well standard plates. The deep well plates are incubated and centrifuged before 250 microliters is transferred from each well of the deep well plates to corresponding wells of black, flat-bottomed 96 well plates. The 3 plates are then read on the fluorometer and the resulting data are analyzed. Ultimately, potential soil extracellular enzyme activities are determined by the rate at which fluorescent dye is released from the substrate by an enzyme-catalyzed reaction, where higher fluorescence indicates more substrate degradation.
The main advantage of this technique over existing methods is the ability to measure enzyme activities in a large number of samples by taking advantage of the deep 96-well microplate format. We first had the idea for this method when we saw the opportunity to increase sample volume compared to standard microplates by adapting other protocols to the deep well plate format.
The implications of this technique allow us to better link microbial physiology to ecosystem scale processes. Visual demonstration is critical for this technique because there are many nuances in sample handling that are not easy to follow in a written procedure.
This method can help answer key questions in the fields of microbial ecology and soil biogeochemistry, such as how do soil microbes respond to changes in their environment, and how do their physiological responses affect critical ecosystem processes, such as decomposition and nutrient cycling.
Though this method can provide insight into decomposition of soil organic matter and nutrient cycling in terrestrial ecosystems, it can also be applied to marine and freshwater.